environment. And it does this by the expansion and contraction of a protein filament that runs through the cell, which is called an axial filament. Now, there are a couple important things that you need to understand about Treponema pallidum. One, it's an obligate anaerobe, so it dies very quickly when exposed to oxygen. Secondly, it's highly motile, and it moves not by flagella, but in a rotation manner. It's one of the few bacteria that will penetrate intact skin. This organism can actually bore its way through your skin to get into the deeper tissue. It is not grown in the laboratory. We cannot culture this organism. The result is that when we go to identify the person that has this illness called syphilis, it's not going to be based on growing the bacterium organism, but by looking at the serology. It's very delicate, anaerobic, highly motile, can't be cultured in the laboratory. These are all important characteristics of this organism. Since you can't grow it very well, and it doesn't stain, or when you stain it with a gram stain, you can't see it because it's very long and very narrow. Here you can see it's a very long, narrow organism. And one way in which to see the organism is to use a type of microscopy which is called dark field microscopy. And with a dark field microscope, what you have is a special condenser. And the light does not pass through the sample, but rather is oriented to bounce off the sample. So that with a dark field microscope, the background is black, the microbe appears white in color, and it's unstained here. And this is one way in which the organism can be visualized. You can't see it if you try to do a gram stain with it because it's too narrow. But with a dark field microscope, you're able to see it. And you can actually aspirate the chancre during the primary stage of this illness and see it if the person would present at that particular time. Now here's an example of the clinical course that you're going to go through with this disease that we call syphilis. When you have contact from an infected host to a susceptible host, the organism leaves the lesion in the infected host and touches the skin of the susceptible host. Now, the chancre here that you see represents where the organism entered the body. It is highly infectious. It contains billions and billions of treponemal cells. The thing about the chancre that's so fascinating is that it doesn't persist for more than a few weeks. It disappears. And it's said to be painless. Now that doesn't look painless to me, but it may be that the nerves are affected, and here it's in an area where one can see it. Often the chancre could be in an area where it is not visible, particularly on a vaginal wall or on the external genitalia, and it will disappear. But coming in contact with it is going to be very important in transmitting the infection. Here's another example of a primary lesion. So the first lesion that develops, usually 1 to 3 weeks after contact with an infected host, is going to be called a chancre. The chancre, again, represents the portal of entry, and during this stage of syphilis, it is easily transmitted from one person to another by coming in contact with that. Another example of a chancre, here's a chancre here with someone who had anal-rectal intercourse. So wherever this organism enters the body, it's going to leave this or cause this primary lesion to develop. Here's another chancre underneath the lip of the individual. Now, again, the chancre is going to disappear in about 6 weeks. So 1 to 3 weeks you take to get the development of the sore. The sore persists for a couple weeks and then goes away. And if it goes unnoticed or it is not painful and the people don't understand what's going on, what will happen is they think there's no other problem. But what happens is that the organism leaves this site, goes into the regional lymphatics and then throughout the body, and you go into what's called the secondary stage of syphilis. This occurs about 2 months after the primary stage. And the characteristics of the secondary stage is a rash that develops over the body. Often it's very intense on the palms of the hands, the soles of the feet. The rash will develop. Also, lesions will develop in the mouth of an individual and on the other mucous membranes. So they can get oral lesions, vaginal lesions, rectal lesions. And during this stage of the illness, the disease is also highly infectious. So during the primary stage, secondary stage of syphilis, this disease can be transmitted from one individual to another. Here are some pictures of the rash that might develop. We'll talk about something like this a little bit later. These look like they resemble what are called gummas. Here's some more of the secondary rash that develops. Now this also disappears. Everybody who gets the primary stage goes on to get the secondary stage. And then what happens is that the body undergoes a biological cure for most people, so that only 25% of the people that get the secondary stage are going to go on to get a later stage of syphilis, which is going to be called tertiary syphilis.
How many?
About 25%. Here's some more hair loss. There are some other symptoms, but the rash is the primary effect that occurs during the secondary stage of syphilis. Now, many years later, after the secondary stage has long disappeared, 1 individual out of 4 will go on to develop what's called tertiary syphilis or latent syphilis. This occurs 20, 30, 40 years after the infection. The organism appears to be sequestered in some parts of the body and will trigger an immunological response. It's believed that the lesions that develop in tertiary syphilis are a delayed type of hypersensitivity mediated by the immune response. The places where the organism causes the damage are primarily the cardiovascular system, with the development of an aneurysm with the aorta. An aneurysm is a thinning of the vessel wall, and you don't want to have an aneurysm high up in the aorta, because under pressure, if it ruptures, and the individuals will die quickly. The organism will also affect the central nervous system. So not only the cardiovascular system, but the central nervous system are the primary sites where tertiary syphilis takes its effect. If it affects the optic nerve, it's going to cause blindness. It will often affect regions of the brain that will lead to dementia and insanity. Paralysis. These are all symptoms of tertiary syphilis. Blind, paralyzed, and insane for your tryst at 20 years old, and now you're 50 and you're in a mental institution. There is no treatment for tertiary syphilis. The organism isn't around anymore to kill, so you're not going to be able to treat this. You treat the symptoms. The disease is not transmittable during tertiary syphilis, only during the primary and secondary stages of the illness.
Now, can they find it in between secondary and tertiary phase?
Yeah, and we'll talk about how the diagnosis is going to be made. The diagnosis is going to be serological. Often you'll form what are called gummas, G-U-M-M-A-S, and what these are masses of benign tissue. They look like cancers. Here you see a gumma that's developed at the surface on the skin, but you'll get lesions like this that develop on the bone and the other internal organs of the body. Here's one where the aorta had occurred. One of the major places the cardiovascular system is susceptible to tertiary syphilis. Here is the organism that's been stained with what's called a silver stain from biopsy material from someone's brain. And you'll see the treponemal cells in the nervous tissue here throughout the brain. This was done following the autopsy of the individual. Now, syphilis can be transmitted from mother to child. If a woman becomes pregnant and contracts syphilis, the organism can pass through the placenta and attack the central nervous system of the child, and we call this congenital syphilis. And congenital syphilis will often have severe effects on a newborn. It may range from such things as a cleft palate. Sometimes they develop a unique pattern with their teeth where the teeth are notched. This is called Hutchinson's teeth, that is characteristic of an individual that had congenital syphilis. These children may be neurologically impaired. They may be stillborn because of the organism attacking the central nervous system. Here's a cleft palate with an individual who is born with congenital syphilis. Okay, let me see if I have another. I think I have a whole other thing here. Might have some different pictures. We just saw these pictures, I think, so that's not it. Let me go back and look at about this one. Do you have any question about Treponema Pallidum before we get into the diagnosis of this? Let's see. Here is, and I just looked at my last morbidity mortality weekly report, and it was about 11,000 cases of syphilis in 2010 in this country. But you can see what's happened here in the early 1940s. Look at 1945 or so. Look at the number of syphilis, almost 68,000 cases a year. You see a high incidence here between 40 and 41. And then why do you think this number of cases went way, way down into beginning about 1945, 46? Yeah, the advent of penicillin being available. And penicillin was used in the treatment when we talk about treatment. And you can see the distribution of this. You had much more of this. You happen to live in an area where there's not a lot. The Tuskegee study, which was done in Alabama, and you'll see the results of that study a little bit later on. This is some of the material that I've already talked to you about. It's transmitted during the primary and secondary stage of the infection. It cannot be cultured. We call such organisms obligate parasites, and it can be transferred by, it's like gonorrhea, direct contact disease.
So obligate parasite means that it can't be cultured.
That means, yeah, it's not going to be able to grow by itself.
You say obligate parasite?
Pardon?
You say obligate parasite?
Obligate, yes. Now, here's the dark field. Now I showed you a chancre. If you extracted a chancre with a syringe and looked at it under the dark field microscope, you could diagnose the condition this way. Often, however, the person doesn't present because it goes away or it may be unnoticed. So that the way in which it's eventually going to be diagnosed is going to be through a process called indirect immunofluorescence. And we'll talk about the fluorescent antibody test for this. This is just, let's see if there's any pictures. Okay, the primary stage, 10 to 90 days, usually about 3 weeks. The primary lesion is called a chancre. The organism will be transmittable during this stage and can then invade the regional lymphatics. We talked about this. Here again, here is a chancre there and a chancre associated there. And again, if it's unnoticed and doesn't hurt and goes away, the individual thinks there's no problem associated with it. The primary lesions heal, the organism goes systemic, and then a few months later you start to get the swollen lymph nodes, the rash, you may get a candida infection, and again during the secondary stage it's going to be communicable. And here's some of the rash type things that we talked about. The latent stage of this, again, only 25% will do this, although the people who have the primary and secondary stage will have a positive serology for the rest of their lives.
Now, can you get it from the rash, or does it have to?
No, it's not from the rash. There'll be lesions on the mucous membranes of the mouth, the rectum, the vaginal mucosa, and just touching those. This is how dentists would acquire syphilis, working in an individual's mouth that had a syphilitic lesion. They would get it this way. Tertiary stage, again, you have neurosyphilis, cardiovascular syphilis. Gummas are the primary symptoms. You don't communicate the organism at this stage. Here's some examples of things that we just talked about. I think before I talk about this, it'll be easier to write on the board than to try to explain this from a slide. So I'm going to end the show here. And this and try to talk about the diagnosis of this. Since you cannot culture the organism, the way in which the disease is going to be diagnosed is by taking blood from a person and looking for antibodies that are produced. And the first test that's done is a screening test. And the screening test is going to be like the time test for TB in that you're looking for people who are negative. And people who are negative, you throw away. And that way you don't have to look at that further. If you get a positive result, then you're going to have to follow that up. Now, the first test that's going to be used for the diagnosis here, diagnosis of syphilis, is going to be a test called the Vdrl test, or one called the RPR test. And as I mentioned, when you talk about the diagnosis of syphilis, there's lots of acronyms. Vdrl stands for Venereal Disease Research Laboratory. And this is where this test was developed. And it was designed to replace the older test, which was called the Wassermann test. The Wassermann test was a complement fixation test to detect syphilis. Now, in the Vdrl test, what one does is to take blood and you let the blood clot, and then you centrifuge this, and what you're left with here is the serum. And you add this serum to a synthetic antigen, or rather you add the antigen to the serum. So here we could have the serum, and to the serum we're going to add an antigen. The antigen that is added is a mixture of several different things. One is a material called cardiolipin, then a fatty acid called lecithin, and then cholesterol. CHLORESTEROL. And this is the synthetic antigen. And what will happen in a positive test, when you add this synthetic antigen, you get clumping of the antigen. You get a clumping reaction. In a negative test, there is no clumping that occurs here. Now, in the Vdrl test, the antigen that the antibody which is detected, the antibody detected is not from T. pallidum. And what happens when this organism invades your body, it damages your cells, and your cells then express antigens that are recognized as foreign by the immune system. So what will happen here is that the T. pallidum will damage host cells, and then the host cells express an antigen that will react. These host cells will express an antigen and cause an immune response, to the damaged host cells. The antibody that is produced is called a Regan antibody. So you don't detect an antibody specific to T. pallidum with this, but rather you detect an antibody that you make in response to your damaged host cells. And this would constitute then a positive Vdrl test. What you look for are people that are negative, because then you don't have to do anything further with them. So this is a screening test. Because this is a little bit tricky to do and sometimes difficult to visualize, this Vdrl test in many places has been replaced with something called a rapid plasma Regan test. And it's based on the same principle, except you have the synthetic antigen on charcoal beads, so that what you have here, you put the serum on the slide, put a drop of serum, and then you add the charcoal beads with the synthetic antigen, and if the beads clump up, it's just easier to see, and this would be a positive test. Now, if the Vdrl test is positive and the RPR test is positive, you then go and do a second test, which is called an FTA test, or a fluorescent treponemal antibody test. So if this is positive, if RPR or Vdrl, they're positive, you then are going to go do a test called the Fda test. And this stands for fluorescent treponemal antibody test. So here we have the fluorescent treponemal antibody test. Now, in this test, what you're going to do is you'll have a microscope slide here, and we'll have a Vdrl positive serum and a Vdrl negative serum. And the first thing you'll do will be to fix Treponema Pallidum cells to the slide. Now, I told you where you get the Treponema Pallidum cells. They get it by growing the organism in male rabbits. They castrate the rabbit and isolate the Treponema Pallidum cells from the testicular tissue. You buy that in a little vial, and then you fix it to the slide with methanol. Then to this, you're going to add the serum from individuals here. And you would use actually two different slides for this. If the person has truly been exposed to T. pallidum, there's going to be antibody associated with this. So what we'll do here is we'll take our T. pallidum and make it much bigger. And now when you add the serum, if there is antibody that's specific to the T. pallidum, here's the Treponema Pallidum. Here in the Vdrl negative test here, we'll have this. There'll be no antibody to bind to it. Now, you can't see an antibody attached to a bacterial cell. An antibody is a protein. What they next add is they add a reagent which is called fluorescein conjugated anti-human globulin. So, fluorescein conjugated, and it's just a bunch of words here, fluorescein conjugated anti-human globulin. Now, anti-human globulin are antibodies that will bind to human antibody. If you take human gamma globulin and put it into a rabbit or a goat or a horse, you take human gamma globulin, which is a mixture of human antibodies, and you put that into an animal, you inoculate this into an animal, inoculate, say, into a rabbit. The rabbit is going to see human antibody as a foreign protein, and it will make antibody to the human antibody. So what's going to happen now here is that the rabbit will make antibodies that can bind to human antibody. So now we have an antibody that can bind or recognize human antibody, and to the heavy chains of that antibody, they attach, that's what conjugate means, they attach a fluorescent dye. So they put a dye here, which is fluorescein. And now you add the fluorescein conjugated anti-human antibody to the slide that's been reacted with a serum. And what's going to happen here is this antibody is going to attach to the antibody that's attached to the Treponema Pallidum cell. And now when you look at this, you put that slide here, and we'll make this the slide. Now you put the slide on a microscope, and instead of using white light like you use black light. And using ultraviolet light, what's going to happen is that this dye is going to fluoresce. So that in a positive Fda test, what's going to happen here is that you'll see the treponemal cells fluorescing all over the slide. In a negative test, you won't see anything because there was no antibody for the fluorescein conjugated anti-human globulin to attach to. So if the Vdrl test is positive, then this is followed by an Fda test. And one of the reasons you have to follow, and I'll get to your question in a minute, is that with the primary screening test, there are things that are called biological false positives, or BFP reactions. If you had certain immunizations, you had an immunization, for example, for smallpox, and I forget all which immunizations, or you just had mononucleosis and then had an Fda test, you could end up having a Vdrl test. You could have a false positive reaction. So that to confirm the screening test here, you have to follow it with the Fda test. This indirect immunofluorescence is very similar to the ELISA test that we talked about with the diagnosis of AIDS, except instead of having an enzyme on the antibody, they have a fluorescent dye on the antibody. You had a question.
Can you say what you said about the positive and negative test again? It binds to the fluorescein binds to the antigens?
What happens here, and you buy this reagent. You don't go and what happens is that we'll just go through the whole thing. Let's suppose we don't know whether you've been exposed or not. I take some of your blood, I let it clot, I centrifuge it, now I have some serum. I take some of that serum and I add the charcoal beads with a synthetic antigen and they all clump up. That suggests to me that you've been exposed to this organism. That's a positive RPR test, or if we did it with a slide there, you have a positive test. Then we would have to follow that up because you could have had some other viral infection that caused that reaction, because what you're detecting in this test is not specific for Treponema pallidum. It's recognizing cells that have been damaged by some infectious agent, and what we're really looking for are the negative people. If you were negative, we wouldn't even have to bother with you anymore. But if you turn up positive, then we're going to have to follow this with a more specific test. So we're going to take then Treponema pallidum that we have in a little vial that we got after they've isolated it from the rabbit, and we fix the Treponema pallidum to a slide here. Then I add your serum again to the slide. If you have antibodies to that, the antibody is going to bind to the Treponema pallidum. But I can't see that until I get another reagent that will allow me to recognize an antibody that's attached to the Treponema pallidum cell. And that other reagent is called fluorescein conjugated anti-human globulin. And what all that means is it's an antibody that recognizes human antibody that has a dye on it. And then when you look at it under the fluorescent microscope, the cells will fluoresce a yellow-green in color, and the background will be black. And that will confirm the diagnosis that you had syphilis. We can't culture the organism. If you present it with a primary lesion, that lesion could have been looked at under a dark field microscope. But more than likely, you're not going to do it because it's going to go away, and you don't, or you may not even know you have it.
So if the antibody recognizes the fluorescein conjugated.
The fluorescein conjugated antibody here recognizes the human antibody, the antibody that you had in your serum that's binding to the Treponema pallidum. Okay, once the diagnosis is made, treatment is available. prior to 1945, the only treatment was toxic metals like mercury. And they used to use mercurials to treat individuals with syphilis, but they were not very effective. Really, the control of syphilis was achieved through the use of antibiotics, and penicillin for many, many years is the antibiotic of choice. It's a whopping dose of penicillin. It's 4.8 million units, 2.4 million units in each cheek. And we're not talking about your cheeks here. We're talking about your gluteus maximus. If you are sensitive to penicillin and they put that much penicillin in you, you wouldn't get the needle out before you'd have a problem. So there are other antibiotics. I don't know whether erythromycin could be used, but you couldn't use the penicillins if you were sensitive to this. Prevention is simple. You avoid contact with an infected host. How do you know who's infected? I have a kit for $19.95. If you buy 2 of them, no, I'm only kidding. I have a kit for this. So what they tried to do was use barrier methods to prevent the transmission of this illness, like gonorrhea, and this was the use of the condom. Obviously, if you're having 300 or 350,000 cases of gonorrhea a year, you're not using condoms. So the organism can be transmitted in this way from one individual to another. And you have, as I mentioned, about 12,000 cases a year. Syphilis is a reportable disease. And we have we have individuals that are called venereal disease investigators. And I don't know whether you've ever seen any of these pedigrees. They're really interesting when you have an individual who has been treated for syphilis, their name is given to individuals in the state who are to follow up on all the contacts that they have had. So if my friend Anthony there, who's diagnosed with syphilis, we would find out that Anthony had contact with Fred, Bill, Mary, John, and whoever. And then they will go and follow up on all of those, and you get these wide pedigrees. And I had a student one year that was a VD investigator for, I think, 5 or 6 counties. One of your counties, maybe even Clay and stuff. And he would tell me, it's pretty risky going up to a farmhouse in rural Wayne County, knocking at the door and wanting to see the guy's daughter because she's been reported to have contact with so-and-so. And he said it was not the easiest job. It sounds like it'd be easy, but it's not. But it is a reportable infectious disease. Do you have any question about syphilis, gonorrhea, streptococcus, staphylococcus? On Monday, I'm going to show you the Tuskegee Experiment, and in the early 1930s in Alabama, the federal government became involved in a study involving the black males in rural Alabama. They promised that they were treating them for a condition called bad blood, but it wasn't bad blood that they had. It was a euphemism. It could mean many different illnesses. They took elaborate steps to not treat these individuals, and they did it for a variety of reasons. When you hear the white doctor who was one of the participants in it, his excuse was really pretty shallow, that he was trying to learn more about the disease and everything else. And this is one of the things that came out of the study. By letting these people go to autopsy, they would learn more about the clinical course of the disease. So you get to see a little bit about that particular study. Then Wednesday, remember, we don't have class because do you assess anything? Do you go over to the student center? They give you pizza and beer and stuff and ask you to fill out questionnaires or something?
Surveys online.
Pardon?
They have surveys online.
Surveys.
Yeah.
Did anybody answer them or
Yeah.
do them?
You can go somewhere too. They have those
I know people have told me where to go lots of places. What do you mean? You can go,
For like,
do you go to your classes?
I
And
just know my roommate's a CD major, and for CD majors, they have. Like,
what's CD?
Communication disorder.
Oh, okay.
And they have like a specific room where you can go, and they have like specific times to take surveys.
And you write on the wall what you think
They
about?
have like sheets of surveys and
Yeah,
things.
okay. We have our department for, what is it, from 9 o'clock to 4 o'clock. And I may just go there to be an asshole. What did we do for 35 years when we didn't have these surveys? All you have to do is look at the graduates that you turn out. I mean, if they've turned out well, some of them have. Do you wear that shirt around? Where are you now? What school are you at?
I'm still here.
You're still here.
I graduate in 6 weeks.
You graduate in 6 weeks. I may carry the torch in. You know? No, I'm not even going to. I've been to graduation once. I have a disposable gown that my kids would wear at Halloween. And anyway, I will see you on Monday. Have a great weekend. Hey, tell Ebony I was asking about her. I know. Should have been quiet where you were sitting. I didn't mean to single you out, or I was just using you as an example. I saw my grandson, his name was Anthony too, so. Of course, he wears a diaper. I didn't. Okay.
So are you
So
really?
you graduate.
Yeah, I graduate in May.
Why are you going to, is it UAB?
Yeah. It's kind of a weird story how I got there. I applied to the internships because that's usually what you do.
Yeah, they're hard to get.
Yeah, I know. So I applied to 4 of them, and then I got this email from a professor. It's one of those mass emails, and it was just like, I guess their department sent this out and said if you're one of the top students that apply, like you get free tuition and health insurance and fees paid. And stipends.
They have an accredited program.
What?